All right, next lecture.
This week, we're going to go all the way from mutations and evolution to next generation genetically modified organisms, including food. So let's get started right away. We'll even touch upon things like variants and who knows what else. Mutation, bug or feature. Of course, we know about mutants, right? I read about this thing in the Marvel universe. Mutants are humans born with a genetic trait called the what? Why are mutants born with an X gene? Could you bioengineer people to have the X gene? What about mutants not in the Marvel universe? How about the reality, the real universe? Well, in nature, you can think about what happens as an organism makes another organism, makes another when a parent has a child, has a grandchild, has a great-grandchild, and so on and so forth. Whether this be Homo sapiens, a human, or a bacteria, or a plant, what happens when one generation begets or reproduces and makes the next generation, and so on and so forth. Underlying this process can be spontaneous changes in the genetic material, which we'll talk about in detail. The genetic material could be DNA, for you and me, or it could be RNA, like the coronavirus. If there are spontaneous changes in the genetic material encoding the living systems, then maybe the living systems can change themselves. And so mutations are spontaneously or randomly arising changes in the genetic material encoding living systems. A lot of mutations, because they don't matter, at least not immediately, some mutations have no immediate impact on the so-called phenotype, the form or behavior of the so mutated organism. Oftentimes you might refer to these mutations as silent mutations. They don't seem to do anything. But of course, the genetic material is changing, so it might do something later. Other mutations have profound and obvious changes. Here's an example I found from a neat biology class. Got a cat on the left, looks like it's a lion. No, it's a tiger. It's got stripes, it's got orange fur. In the middle is a mutant, still got stripes, but the fur is no longer orange. Something has changed in the animal's genetic material such that the pigmentation of the fur is different. On the far right, we see another mutant. It's albino. There's no stripes, no color of any form, right? So some mutations have obvious changes. That's in nature. But how about in bioengineering? Well, in bioengineering, if we're clever, we can create systems where there are random changes bioengineered into what the living system's doing. And then if we do that right, we might get very interesting tuning or engineering of the resulting properties of the living systems. It's kind of unbelievable. Believe it or not, you're looking at neurons in the hippocampus of a mouse brain. That's why I've got the brain, the cartoon character on the right. And scientists have put DNA into the individual neurons designed to produce different colors in different neurons. But what color any individual neuron makes is not hard-coded. It's not prescribed. It's the result of a random process where through clever bioengineering, there are spontaneous or random changes in the DNA at very high frequency as this brain is developing. And thus, as a result, each individual neuron gets a different dosing of different colors, and you get this beautiful image where you can clearly discriminate between one neuron and the next as the brain's wiring up. This is called a brainbow. And it's just such an amazing demonstration of how you can use clever up. And really, if you're excited about this, check out the URL down there and learn more about it, the brainbow. Okay. Well, back to basics. Where do mutations come from in nature? Or stated differently, like, why do they happen? Do they have to happen? And I want to start exploring this question with the macroscopic. Like, let's imagine you went to a museum, you brought some paint and some canvas, and you wanted to copy a painting in the museum. It's a little bit strange, but maybe you could do it, like this person here. So this person is looking at something and then through a physical process of painting, making a copy of the first painting, right? And we can think about this and just what's going to happen here. Maybe we could say the following. Whenever one object is copied by a physical process, there's a chance that the resulting copy will not be exactly the same due to one or more random differences. Like trying to copy it, but maybe I couldn't figure out exactly, how the hair was on the person in the first painting. I did my best, so maybe I couldn't decode the first painting. So even if I was a perfect painter, I wasn't quite sure what I was seeing. Or maybe I could perfectly interpret the first painting, but my hands were a little shaky. Oops, made a mistake on the ear. Right. So whenever you have a physical process that's copying something, there's a chance, random chance, that you might get a change in the offspring, if you will. I want to test you and test your thinking by giving you just a case study of copying. And okay, so let's take a look, or take a listen in this case. So here's a song. It's that's a number one hit from the late 1960s in the United States in a genre called bubblegum pop. I don't know if I'd recommend it, but let's listen to the hit. Now here's a copy of this song by Bob Marley and the Wailers just a little bit after. So is the second song a mutation of the first song? Of course, it's a cover song, right? The original and then a cover song. But going back to the preceding slide, is Bob Marley and the Wailers version, which I prefer, nothing against the original, but like this sounds like more interesting. Is it a mutant? Think about that. For me, I'd say no, because the resulting copy is not produced via a random process, right? A mutant of the first song would be you're making the physical copy directly, and like maybe you got a note wrong or something changed, but it wasn't this purposeful, conscious, intentional derivative or cover, right? So take care when things are getting copied. Sometimes they're mutants, and sometimes they're just cover songs, right? So trick slide here, but think about it. Okay, let's bring this back to biology and bioengineering. Here we've got a whole slide full of A's, T's, C's, and G's. Can you guess what it is? How many letters are on here? 1, 2, 3, 4, 5, 6, 29,000, almost 30,000. It's amazing how many letters you can get on one slide. Turns out this is a DNA representation of SARS-CoV-2, the coronavirus, which is normally the T's would be used because it's RNA, but this is the DNA version. Here's the whole genome. If you got infected with SARS-CoV-2, this is the thing that's reproducing inside of you, at least an abstracted, representation as the genetic sequence. So I want you to, right now, get a piece of paper and quickly copy the sequence. Go. Are you done? Did you make any mistakes? Of course, it's impossible for using Pencer, but somehow, when we think about mutation in biology, at the core of it is random changes in nucleic acid sequences. And I hope if you seriously confronted the challenge of quickly copy this sequence, that's what the molecules of life are going to have to do with perfection if we didn't want random changes, if we didn't want mutation. Ridiculously difficult or challenging it's going to be to copy these strands of genetic material with zero random changes. Well, let's look at the molecules that actually copy the SARS-CoV-2 genome, at least a representation of it. So this is something we've seen before in terms of structural biology. In this case, we're looking at what's called the RNA-dependent RNA polymerase. Normally, when you think about copying of genetic material, we're thinking about copying of DNA, so you have a DNA polymerase, but some viruses have their genetic material stored as RNA, not DNA. So to copy RNA, you need an RNA-dependent RNA polymerase, which is a mouthful, I'll admit. And what we're looking at here around the outside of this core barrel are the proteins that comprise the RNA copying machinery that are going to replicate this entire sequence. And hopefully, we wanted it to have no mutations. They're not going to make a single mistake when they copy all this sequence. Right down the middle of this, in red and blue, is a representation of an RNA being copied, right? And then all of this is how this one, and then we're just going to rotate the whole thing 90 degrees. And so now you can see the RNA that's being copied. The template is in blue, and then the duplication or second copy of the genome is being made in red and sort of coming out from right to left. Now, how this machine is actually working, this is just a static image, and all the atoms are kind of hidden away in these ribbon diagrams. But somehow, this is the business. You know, like this is the molecular machine that's copying the virus genome that's causing the pandemic. Let's look at an example of a DNA copying machine and see if we can develop a little bit more empathy for what must be happening inside a cell. Again, just on background, SARS copying itself is RNA copying RNA. This example, a beautiful animation from Drew Berry and colleagues, is a DNA copying DNA. And what's shown here is a strand of DNA coming in on the left, and then two strands of DNA proceeding off to the right. And the spaceship-looking thing, of course, it's a molecular complex. That's the set of molecules, the DNA polymerase that's copying the DNA. And here's the business side of it, where you've got a single strand of DNA coming in and a double strand of DNA going out to the right. And right here, we're seeing the individual bases being copied somehow. An A being matched up with a T, a C with a G with a C, a T with an A. And you know, you can kind of get a sense of it, but of course this is super slowed down. And in reality, all of this is happening, you know, in a human cell, you know, maybe it's moving at 50 letters a second. In a bacteria or virus, it could be 1,000 letters a second. And for this to work, you know, like randomly the bases are appearing and the poor polymerase has to decide, I need a G, is that a G or a C? And it can't look, it's like somehow electrostatics and physics, right? It's like, is this process going to be a perfect process? I mean, no. You know, like I can hardly copy anything when I'm thinking as hard as possible about it without making a mistake. How are these molecular systems going to copy millions of base pairs, tens of thousands of base pairs without making, it's just not going to happen, right? It's actually remarkable they're as accurate as they are. They might make a mistake 1 in 10,000, 1 in a million, 1 in a billion, depending on how much, you know, proofreading and error correction there is. All of this is done at the molecular level, but the key thing is this copying process, as good as it is, isn't going to be perfect. And because it's not perfect, there's going to be random mistakes throughout what's going on. So what happens if a polymerase, a DNA polymerase or an RNA-dependent RNA polymerase makes a mistake? Depends. A lot of the time, it's silent. So it doesn't make an appearance on the broader stage. But if it has an impact, if it impacts phenotype, if it impacts what actually happens functionally, at the limit, you know, by the way, it's not if it makes a mistake. Remember, it's when a polymerase makes a random error. You don't know where in the sequence it's going to be, but it's going to be there randomly. So what happens? Well, it depends. Is it silent or not? At the limit, it's not silent. The opposite of is like, it's on the news, right? And we've all been living through the consequences of these random mutations, some of which are not silent. And they have, in the case of the SARS-CoV-2 pandemic, significant impact on how the pandemic's progressing. Variants, you know, the Omicron variant, the Delta variant, this language we're seeing in the news is traceable back to the physical process of copying nucleic acids, and the fact that that copying process, as good as it is imperfect. And so we get spontaneous changes that result in mutations, and some of the mutations have significant consequences for how the virus infects, and that's a variant, right? So now hopefully you're a little bit more motivated. It's not just the X-Men, it's like, oh, it's the pandemic. It's what evolution does. So let's see if we can make sense of this. And remember, we're practicing throughout the course, parsing the research literature. This one's going to be a challenging one. All right, so let's look at the title first. This is a paper that came out in February of 2022. So this paper is 13 months old, right? SARS-CoV-2, okay. Omicron variant, I've heard Omicron, it's a variant. I guess that means it's a mutant. Well, it's a mutant that matters, so it's a variant. And then antibody evasion. Antibodies are part of the immune system. Evasion, something's evading part of the immune system. And cryo-EM, jargon, structure of spike protein ACE2 complex. Spike protein's a part of the virus particle. ACE2 is the part of your cells that normally would regulate blood pressure, I think. But it happens to be the part of the surface of your cell that the virus binds to and comes in on. It's so interesting. So now let's go to the abstract. The newly reported Omicron variant is poised to replace the Delta variant as the most prevalent SARS-CoV-2 strain variant across the world. Cryo-electron, okay, so that's cryo-EM microscopy, right, got it. A microscope, a fancy microscope, analyzing the structure or shape of the Omicron variant spike protein. So how the mutants cause the spike protein to have a different shape. An analysis of how the shape changes the complex with the human angiotensin converting, aka ACE2. Again, the receptor that the virus binds to that's normally doing blood pressure stuff, but in this case, it's how the virus gets into our cells. Reveals salt bridges. Sounds like I drive a car over a salt bridge. Can't be what that's about. Hydrogen bonds, mutated residues. Mutants are appearing. Good. Mutated residues, arginine 493, serine 496, arginine 498. Whoa, I told you this is going to be a hard abstract. I'm just going to stop here. And I'm like, this looks interesting. A microscope is revealing how things are physically shaped in relation to each other. And there's mutated residues, things like serine 496. All right. We're going to come back to this, but I want to just pause and make sure everybody's following what's going on between the spike protein and this thing called ACE2. And so this is just not from this paper, but just on background. What's the life cycle and how does SARS-CoV-2 work and transmit? So if you're getting a SARS-CoV-2 particle coming into your body, on the virus particle itself, on the outside are these things called the spike proteins. And for the particle to do anything, those spike proteins have to interact with a specific molecule on the surface of some of your cells. This is called the receptor. And the specific receptor that the SARS-CoV-2 spike protein interacts with is called ACE2 or ACE2, right? This thing I've been mentioning has something to do with regulating blood pressure, I guess. All right, so that's just what's going on. And if you had a mutant that is changing the structure of the spike protein, then you might expect it could change how the spike protein interacts with the receptor. And since a lot of the antibodies that your immune system might produce, are basically trying to prevent this binding event, the antibodies are going to be interacting with the spike protein too. And so if the spike protein mutates, then an antibody that might be giving you immunity from infection, perhaps generated by a vaccine or a prior infection, that antibody might not recognize the mutated spike protein, hence a variant that can escape and cause reinfection. So in other words, your vaccine might not protect you from the latest variants. That's where it's all coming from the mutations. All right, so let's go back to this paper, and we can find this figure in the paper, and it's like, whoa, my gosh, this is complicated. But I see things like, I what was it, 496 serine? I'm going to go back and double check. What was that about? Yeah, like mutated residue serine 496. So just remember that, mutated residue serine 496. So what this is depicting is just looking at the physical structure that they're measuring on their fancy microscope and where the mutants are on the structure of the spike protein, right? So I see the number 496 and I see the letter S. And, that's what the physical structure looks like and where that mutant is. It's somewhere, I guess it's out on the tip of the spike protein where it's going to be interacting with the receptor. And so, that's that, and, that's the region that the antibody might bind to. And so, but what does 496S mean? And more specifically, if we go back and look at this, it's like, it's not 496S, it's a G496S. So, whoa, where did all this jargon come from? How are we going to decode it? So let's see if we can figure this out. I'm going to use the Gettysburg Address to quickly explain what the heck things like G496S mean. So
wow. There's
Abraham Lincoln with his top hat. What an amazing photo. And if you know the Gettysburg Address, it starts with the 407 years ago. So the first thing we're going to do is we're going to put a number. We're just going to number the letters in order. So the first letter is an F, and so that's a 1. The second letter is an O, that's at position 2. The third letter is a U, that's at position 3, and so on. And so this set of words has 25 different letters, and we can say, there's an A at position 23, and so on. Okay, good. Now, I'm going to propose some changes for mutants, right? Just changes. So, for example, the letter C that is historically at position 6, let's change that to a P. Or the letter Y that's historically at position 18, we're going to change that to a B. Okay? And the letter A that's at position 20, right here, we're going to change that to an E. So if we make all these changes, we'll get a different set of words. Let's see, what would those be? Let's make these changes. Instead of 407 years ago, we make these 3 changes and we get, 407 beers ago. So when we see C6P, we're taking the original letter C at position 6 and replacing it with the letter P, and so on. And that's what's happening in the spike protein, but with a different alphabet that we're going to have to introduce on the next slide. But quickly now, you can hopefully see this type of jargon and how it works. It's a coding system for taking one letter and turning it into a different letter at a particular position. So what's the language? What's the coding table? G496S. What does that mean? It's not going to be the letters of the English alphabet. These letters refer to the single letter abbreviation of the amino acids, the 20 amino acids that make up proteins. From alanine, which is abbreviated with the single letter A, or the 3-letter ALA, arginine, asparagine, aspartic acid, cysteine, glutamine, all the way down to tyrosine, which is abbreviated with the single letter Y. That's awkward. Well, that's because T was already used, just above for threonine. Anyway, these 20 letters refer to the 20 different amino acids that comprise proteins. So G496 is, well, we had a G, which was a glycine, and we're going to turn it into an S, which is a serine. And we're going to do that at position 496 in the spike protein. Just to restate that and write it out, at position 496 in the polypeptide chain of the spike protein, we're going to replace the original G, or glycine, with an S, or serine. And so I was able to find the spike protein sequence online at the website at the bottom, and here are the single letter abbreviations for all the amino acids making up the SARS-CoV-2 spike protein, starting with an M and ending with a T. And if we find the G, 496 letters into this thing, G496S means we're replacing that G with an S, and that's it. right? So this is the G496S mutation or variant found in, the mutated spike protein that's taken over the pandemic. Now, to be careful, these changes in the protein sequence are not mutations per se. They're the result of an underlying mutation in the nucleic acid sequence. So what do we mean by that? RNA or DNA encodes triplets that are then decoded by the ribosome and the tRNA to make proteins. And there's a lookup table, the famous genetic code, that tells you how to do the decoding. So each triplet in RNA, in this case, will result in one of the 20 amino acids or a start or stop sequence. And so glycine, for example, or G, is encoded by any of these four triplet codons would get you a glycine amino acid, GGU, GGC, GGA, GGG. And if we have a mutation that's resulting in a serine, we can see that there are 4 options for serine over here, UCU and so on, but also 2 more options for serine over here, AGU and AGC. So if I had a random error in an RNA copying process that caused a glycine on a protein to become a serine, I've got to be moving from one of these 4. But to jump all the way over here to one of these 4 serines, I'd have to change more than one letter. Whereas if I just want to pop up here, like GGU, that's a glycine. If I just change the G to an A on the RNA, I'll get AGU. So just a G to A substitution will move me up this codon table in a mutation, if you will, from a glycine encoding codon to a serine encoding codon. So probably what's happening during the evolution of this particular variant in SARS is in the protein. It's originally encoded by either GGU or GGC, and it's most likely becoming either AGU or AGC. Right. So this funny notation is at the protein level. The mutations themselves are at the nucleic acid level, and you've got to decode it through the codon table here. So that's a lot. We're going to pause there. Reflective questions. What are mutations? Random changes, remember. All right. Why do they happen? Any physical copying process is likely to be imperfect in ways that are random. Is it possible to stop this from occurring? Not absolutely. You can do a lot of correction, but that's going to require energy. And so depending on how much energy you have, right? Okay. Why do mutations matter? Are mutations always bad? If we can't stop mutations from happening, can we stop them from mattering? Sure, if you stop replication by squelching a pandemic, you won't get the mutations. There just won't be enough replication, but there might be other ways to stop mutations from mattering, right? But hidden within this question is a whole bunch of policy topics you can think about, like how urgent is it to get people vaccinated? Could you invent a game or experience if you were talking with somebody who had no idea what mutations were? Like what experience could you invent for them that might help them develop empathy for this puzzle of why do mutations occur in the first place? How's that related to physical copying? One of my favorite ways of exploring this, for example, in a classroom, is to get 10 people in a line and give the first person a written phrase to whisper to the next person and cheer everybody on to, as quickly as possible, get that phrase whispered all the way down the line of people. And the best thing to do is to have another line of people and compete to see who get there first without making any mistakes in what the saying or phrase is. But I'm sure you can come up with other ways of helping people think about where do mutations come from.
Wow.
This week we're going to talk about...
Well, I am feeling a little tired today. Don't have too much to say about that lecture. Can't think of too much. I am feeling a little bit tired. I'm saying that's it for